There's no echo.
Is the screen big enough?
Yes, yes, it is.
I've got it on slideshow.
Yeah, yeah. No, that looks fine.
Okay.
So why don't you proceed, Bob? I think that would be
fine. All right. So good afternoon. I'm Bob Rennie. I used to do anthrax. Now I'm apparently doing.
We used to do it together, but this is more topical.
Right. So, yeah, and the reason we did anthrax because it was perhaps more in the realm of destructive microbiology, right? And so there was discussion about how evil could possibly prevail, and I'm going to debunk that theory here as we go through. So this is really just about what we know and what we don't know, and we still don't know lots, and we think we know some, and then tomorrow it will change with this virus. And so this is SARS-CoV-2, in the popular press called COVID19. It actually stands for Severe Acute Respiratory Syndrome Coronavirus 2. So it's the second of these viruses that have been important. And as you'll see, I'm a professor emeritus in laboratory medicine and pathology. Makes me an old guy. So now how do I change this? Oh God, let me just see. There we go. I got it. So COVID, this stands for coronavirus, identified and called COVID, coronavirus ID, identified in 2019 as a new strain. So when we look at there are only about 7 strains. There's hundreds of coronaviruses, but there's only about 7 strains associated with human infection, and they've been around for centuries. There are alpha coronaviruses that cause mild respiratory disease, like the common cold, among other viruses that do the same thing. And there are these beta coronaviruses, and these are new strains that have been seen in the past 1 to 2 decades. So the first of these was SARS-CoV-1, which was the original SARS virus that we saw rising out of Asia, particularly in Hong Kong. And remember the apartment buildings where there were multiple diseases, and it arrived in Canada by airplane, primarily in British Columbia and in Toronto. And to put it mildly, it was badly handled by public health in Toronto and resulted in a reasonable number of cases and several deaths. But petered out because it didn't spread widely for some reason. The next one was 2012, and this was MERS. This is a Middle Eastern respiratory coronavirus that occurred only in the Middle East. And because there's not much spread and travel out of that region, having been there, there's not much spread and travel out of it, and they shut things down pretty well, and MERS disappeared. And so the newest one, as I said, is this SARS-CoV-2, and it's a new variant of SARS. It appears to be more virulent, and it's better adapted for transmission. And there are multiple reasons for that, but we don't know a lot about that. The adaptation of this virus, like many other of these respiratory viruses, appears to be animal to human transmission, often with an intermediate host. And SARS-CoV-2, as the popular press initially indicated, appeared to be most likely acquired from bats because it's been found in the bats in China and certainly and potentially in other areas. and possibly then through an intermediate host, as it were, which we think of more in terms of parasites, but cats, possibly mink. And you'll remember in the mink, they've destroyed in Denmark about 17 million mink. They destroyed mink in a couple of farms in British Columbia as a precautionary measure to help prevent spread. And the WHO reported in it, I originally said March the 9th, but it was actually yesterday. So my slides are actually up to date. The WHO reported that it was most likely acquired from bats, possibly another animal, but that was it. And the reasons for that spread and transmission likely have something to do with how this particular virus mutates to become more transmissible. So this is SARS-CoV-2. It's primarily it's a single-stranded RNA virus. It's got an envelope. It's got a lipoprotein envelope. It has a nucleoprotein. It's pretty small, actually. It has genomic RNA. It has this spike protein, and the spike protein is the primary surface protein that results in attachment to respiratory cells. And that's been was well recognized early on in the development of the pandemic. And then it has this other membrane protein. So pretty simple virus, but it has the ability to mutate and to be transmitted. And just to remind us all how these viruses get into our host cells. So SARS-CoV-2, and if you look up at the, I don't know whether I can show, is my little arrow showing on this?
Yes, it is, yes.
Okay, so early on, a researcher in China showed that this spike protein attaches to this angiotensin converting enzyme receptor protein on the surface of respiratory cells in humans. And that's a major breakthrough because it also has important potential implications for treatment. And I'll come back to that much later. So all of these viruses and most viruses bind an entry. Once they bind to that ACE2 receptor, they bind and they enter the cell by what's known as a fusion process. So it just is basically an encapsulation of the membrane around the virus, and then it seals it off and allows it to enter the cell. There's then endosome and lysosome tracking. There are things that break open the cell, allowing the release of the viral genome. Remember that this is an RNA, so it has to be transcribed through a DNA process, and there's a polymerase protein and a replicase that this viral polymerase allows the genomic RNA, and remember it's a single-stranded RNA, to replicate, to then produce the transcription for the spike protein, the membrane protein, the envelope. Those are then reassembled on the endoplasmic reticulum. They're assembled, and then they bud, the whole virus then buds out of the cell. And then multiple viruses multiple components of this, multiple whole viruses will be then released from the cell and go on to infect other cells. And so you can see where we need to have our efforts according to preventing either binding or preventing release or preventing the transcription of this virus within the cell. That's how we'll get it dealt with. I just want to do a comparison here because there's another virus which is interesting that we've had pandemics occur. And this is influenza A, and this is structure of influenza virus. Again, it's a single-stranded RNA virus. It's surrounded by a lipid envelope. Oops, sorry. Let me go back. It's surrounded by a lipid envelope. It has two major, quote, virulence factors, a neuraminidase, which is important for binding the cell to the to host cells in the respiratory tract. It has a hemagglutinin as well, which is a hemagglutinating protein, so that also helps it bind. And it has capsid proteins and another nucleoprotein, very similar to SARS-CoV-2, but it doesn't have this same spike protein, and it doesn't interact with the surface of the cell through this angiotensin converting enzyme receptor. And so that may be why influenza is overall less virulent than we've seen with this particular virus. Just want to talk a little bit about this transmission issue, and this is what occurs with both of these viruses. So there seem to be birds, and you'll remember the bird flu, and all of the birds in British Columbia, the chickens and the ducks and everything they destroyed lots of those to try and prevent spread of the virus. And the major one that we've seen before was the swine flu virus that occurred in the pandemic of 1918-19. And so very similar mechanisms for transmission of these viruses from birds or bats or pigs or other animals into man, okay? So not dissimilar, but there are some differences. So the anatomy of this, it's a single-stranded RNA virus. It has a lipid envelope, a lipoprotein envelope. It's has a protein capsid, which is just a surface thing to hold the thing together. It has structural and non-structural proteins, and there are virulent surface proteins, which I've mentioned before into these. And the two major ones for the SARS-CoV-2 excuse me, are the spike glycoprotein and an so-called E membrane protein. And those are important, as we'll see, for vaccine production. So the issue is, why was there such significant transmission in this case to humans? So it appears that the mutations have occurred in the transmission of bats to humans or bats through other animals to humans that have made the virus more transmissible. And we're not really sure what specifically has allowed that to happen, but it may be that it's made it more, that the affinity for this ACE2 receptor has become more important in terms of that mutation. The other component is that there was no prior exposure to this particular virus in humans that we know of prior to 2019. Therefore, there's no innate antibody. So we don't have any, since there's no exposure, humans don't have antibody to this particular virus. Thirdly, it initially occurred in very dense populations. For example, as we know, it appears to have started in this very densely populated area, this Wuhan marketplace. And whether people believe or apparently, according to the thing, a researcher had in China had made an alert that this virus was present, and he was not, it was not really felt that it was that important. And he probably was proven to be right. But those, again, are speculations, because we often don't know what's happened in those kinds of situations. Interestingly enough, since the pandemic started, we've also seen since the start of the pandemic, we've seen acquisition in other animals. So in the zoos in New York, Louisville, and Barcelona, lions and snow leopards have been shown to start coughing. This virus has been isolated and they've all had minor symptoms in recovery. And in those settings, there were no transmissions to humans, even though the workers work in those areas. There have been similar findings in dogs and cats, again, with no minor symptoms and no transmissions. And mink, particularly in Denmark, but we've also done it here in British Columbia, where there are some mink farms. They've had more severe disease. Not many deaths so far as I know. And in Denmark, they destroyed 17 million of them too, as a precaution. So I just wanted to add, this is not the China virus, which you've undoubtedly heard in the news media and social media. So it's a virus that just happened to occur there. And in many cases, for example, with influenza, the viruses that we normally see each year arise out of Asia as well, probably because of the issues of their winters and our summers. And by the time things move, those viruses tend to have occurred more frequently and are more likely to be transmitted across the world. So, secondly, I just want to debunk some of these conspiracies. There's no evidence to support the notion that made prevalent, particularly south of the border, south of the Canadian border, that this was a deliberate attempt to spread this particular virus with a leak from a laboratory. If we go back, most of you will not have been to the lecture on anthrax, but there's reasonable information that, although it's a bacterial infection, that it might have been spread initially directly from a laboratory in terms of one of the outbreaks, one of the original outbreaks of that disease. There is no evidence to this. And there's also no evidence that it was made in a laboratory and then spread initially in specific areas and then released across the world. I've been involved in, we've been involved in other cases, and there are other cases in the literature where people have attempted to make mutations of both bacteria and viruses to try and show that they could be spread, and none of those have been successful. So like many other respiratory viruses, they're present and prevalent in the environment, and the usual likelihood of that infections occur and then disappear. And so we need more than just the virus and its virulence. We need conditions to help move things along in terms of a pandemic. And for example, in the Spanish influenza that occurred at the end of World War I, it's highly likely that there were large numbers of troops, and it's been shown that there were large numbers of troops who were being demobilized and then sent back to North America and to their own countries, taking the virus with them. And many of them were sick by the time they actually got off the boats, because there was no large amounts of air travel. So the point of this slide is that without prior exposure, as we've mentioned before, and now with rapid transit and transport, rapid spread can result in a pandemic. And we see this, there's rapid transit across Europe, there's rapid transit across by trains and planes across all of Asia and all of North America, and nobody thought to shut things down when this started. So that's what results in what we're seeing. So just in terms of perspectives of pandemics, If you go back to about 1350, look at the population. Now, this was the bubonic plague. So this is Yersinia pestis. Again, was a tick and a rat involved in spread of this bacterial infection. 450 million people in the world, and about 40% of them died until they started cleaning up the rats. In smallpox in 1520, there were 460 million people, 12% plus approximate, and these are approximations, died. If you look at the Spanish flu, in which there were a large number of deaths, there were 1.8 billion people in the world, 45 million deaths, attributable deaths, so a rate of about 0.8%. And this is the most recent one. And so these estimates are based on population and on the numbers of deaths up until about the 20 to 24th of March. 2.8 million deaths that we know about. So a percentage rate of about 0.03 to 0.04%. And I put this in the bottom, but there's different estimates of social and economic deaths. And the social deaths are those that have occurred because of lockdowns, suicides, which have increased, economic deaths, people losing their jobs, and a lot of other components of this that probably didn't occur to the same degree. So if you think of it in a larger term, it might be a little greater than that. And it's worthwhile remembering that these pandemics can cause, unless we do the right things, can cause significant both social, economic, and even considerable morbidity and mortality. If you look at global and Canadian cases, so again, about 2.8, there are about 126 million, oh, sorry, keep hitting the wrong button. 126 million cases globally. Remember, this is cases, not population. And deaths about 2.2% of the total that we know about, again, because reporting varies from country to country. We don't hear a lot out of Russia, for example, so we're not sure what those are like. In Canada, the data is reasonably accurate. There have been just under 1 million cases and about 23,000 deaths, about 2.4%. So similar numbers as occurred globally. And in Alberta, and this is just for local, we've had about 150,000 cases and 2,000 deaths for a mortality rate of about 1.3% of those who have acquired known to have acquired the virus. Okay. If you look at Canada as a whole, you can see the dark blues. Quebec, Ontario, and Alberta have been the big areas. That's likely related to population. If you read the newspapers and you listen to the public health people, there are other issues around lockdown or relaxing of lockdown that have led to the seconds or third, possibly the third waves. You can see in highly unpopulated areas, wait a minute, where am I? See, in highly unpopulated areas, there are relatively few cases there. Appear to be more in Nunavut, which is this component here, possibly because of lack of availability of medical care and families and communities that are closely knit and don't have access to either testing or large amounts of testing or adequate control measures. So how do we control this virus? Which is, to me, the most important thing. Masks. Anybody who says that masks don't work is silly. And there are lots of anti-maskers around. Hand washing. Interestingly enough, we talk about respiratory transmission, but there are lots of other examples that we can use that show, for example, streptococcal pharyngitis is primarily acquired because people don't want primarily acquired from the organism being on skin and then people touching their noses or faces and don't wash their hands. And how many times have I been in an airport, even just prior to this, walking into a bathroom in Toronto or Vancouver or Edmonton and going to the bathroom and washing my hands and seeing how many, particularly males, I don't know what happens, who go to the washroom and then just turn around and walk back out. It's just unbelievable. And you don't want to say anything because you'll probably get plowed. But we probably should be saying more. Social distancing has been shown that this virus doesn't, I mean, the modelers show that the spread can be significant up to 50 feet and stuff. And those are all done with artificial modeling. And I think we need to be careful how we look at that. There's actually an interesting, it was a little funny thing on YouTube about beer and masks and a blowtorch. And if you get a chance to look at it, it's kind of cute, but it tends to show that it's hard to actually burn the mask with the blowtorch and volatile aerosols unless you're relative within that sort of 2 meters area. So it's not far off in my opinion. It's important that we test, and we test widely. And as you'll remember, particularly in the United States, where they've had huge numbers of cases, 500,000 deaths. They have a quarter of the deaths of the entire world. They've had a quarter of the deaths of the entire world. That testing was debunked primarily by the previous president, and that tracing is also important. So if you'll notice, even now, which is late, go into a restaurant that are now serving again, and you're asked for your name and phone number, and that's so we can trace, and that's important to try and prevent spread. Closing of public events has been important. You'll be seeing in the hockey that they've had very few cases. There's nobody looking at those games. And then lockdown as the most severe of these, and we're still to some degree into that, and we will be for some period of time until the environmental load, and I use that term loosely, the environmental load in our various areas is low. And what they're using now is numbers of cases in hospital, transmission rates of greater than 1, and numbers of patients in ICUs to try and get a handle on whether or not lockdowns should be relaxed or whether they need to be put back in place. The interesting thing, and I noticed this when I wrecked my ankle, and also from grandchildren, is that there have been no case in Alberta. There have been no influenza reported cases, and there have been more testing this year for influenza than in previous years prior to the pandemic, the COVID pandemic. There is no influenzas. It's also been seen in Australia, where we often see influenza arising. And there have also been as the ER doctor I saw said, there have been almost no other, she hasn't seen a child in an emergency room with a respiratory infection for a year. So something to do either with viral competition or with all of these things that most people have been doing to try and prevent acquisition of the SARS virus. So it's really fascinating. It will be interesting to see what happens to these kinds of viruses, or do we see new ones that we haven't seen once this pandemic slows down and is perhaps over. I want to talk a little bit about testing for the SARS-CoV-2 virus, the considerations. There are a number of things that need to be considered. Timing. The incubation period is anywhere between 2 or 3 and 14 days. That data is relatively poor because we don't know, it's hard to estimate when the virus was first acquired in an individual. Specimen collection. So the collection site, again, the timing, because if you time it too early, the thing will be negative, yet the virus, because of sensitivity of the tests of the sample collection themselves, the sensitivity and specificity of the collection methods, and I'll mention a little more about that in a moment, and what actual targets we're looking for. So the important consideration is the intended use of the test. Do you want to screen or do you want it to be diagnostic? And for example, I'll give you an example, and you may have seen this in the newspapers. The person who started testing in the airports, and an individual arrives from the United States, having been at a meeting, screens negative with a screening virus test in Calgary Airport, goes home and infects all 8 of her family. So again, the sensitivity and specificity of the methods and whether they're being used to move traffic or to actually be diagnostic are important considerations. And how the specimen is collected will primarily determine the likely result. So the major thing are respiratory specimens. The most sensitive of these is nasopharyngeal sampling. But that tends to be more painful for the person being tested. You can also test by throat swabs if they're done correctly. You can use nasal sampling. And so, again, I don't know if you've watched some of the things with these drive-through testing places in the United States, for example, where the tester who may have been given some education on how to test, basically close their eyes, sticks the swab into the anterior portion of the nasal cavity, pulls it out, and I can guarantee you that those tests are at best 70% positive. Versus a mid-turbinate swab where there is some pain, and in both nostrils where the virus is more likely to be lodged. There have been studies using saliva, some people saying that spit is fine, but their studies are poorly done. And there are some new studies, primarily out of Arizona and British Columbia, that show it's called what they call as a gargle and spit. So it's actually throat related, but it's salival produced by a gargle in the very back of the throat and then put into a tube. And these ones have been shown to be actually relatively good, of a similar order of sensitivity as throat swabs. We don't know much about bronchoalveolar lavage. Those are done in hospital and tracheal aspirates, but it's expected that they will be of some value as well, particularly for patients who we already know, and we're looking to see whether our treatments are useful in getting rid of the virus. So those are important, and I just want to show you the anatomy. So this is the nose. This is, whoops, where am I? This is the anterior nose, and that'll take a swab up to about there. So this is the middle turbinate, and this is what we're talking about. That's a long way from the edge of the nose. And then this is the nasopharynx. So you've got to go a long way to get to the nasopharynx. And I can tell you as an example, because I was involved, although didn't acquire the virus from the doctor's curling bonspiel back in a year ago this month. And one of the physicians went back to his hometown. He was coughing. He had anosmia, which is loss of taste and smell. He got an anterior nasal swab, and it was negative. And that same day, probably because he was a physician, he went back the next day, and as he says, they swabbed my brains. So he got a nasopharyngeal swab, and he was clearly positive. So there are a lot more false negatives if you don't do the test correctly. You can also use blood, but it has to be plasma for testing because plasma contains the cells that the virus lives in. You can do feces. There's some indication that people may excrete the virus through feces. You can look at sewage, and this is being done as surveillance, particularly in long-term care facilities, to try and get handles on when an outbreak might happen. And I think we're late for that for this pandemic, but it may be of value as an early harbinger for particular virus spread in outflows of sewage from particular spaces in order to determine the likelihood of spread within those kinds of facilities. And you can also do lung biopsy in very serious cases. So on to testing. So the primary mechanism is to do what we know as nucleic acid amplification or NAT tests. And what the gold standard, as it were, is a quantitative or qualitative reverse transcript PCR test. So because it's viral RNA, we have to use a reverse transcriptase to change the virus to DNA so we can test it, because you can't, unless you do just genomic analysis, which takes too long, you can't find the specific viral RNA. So these PCR tests will look for generally at least two viral antigens, a protein membrane antigen, or the viral RNA to at least two of these antigens, a membrane protein or E protein, and an RDRP gene that's in a different segment of the genome called the open reading frame 1B segment. And I'll show you a photograph of that in a moment. And there are some tests that individuals are looking for 3 coding genes just to be certain. But the primary one is the presence of this E or membrane protein. And you can also look for the RNA, the genome, the segment of the genome that looks for the spike protein as well. So a positive, either qualitative or quantitative test will indicate exposure to the virus, to the SARS-CoV-2 virus. The presence of the virus will also be, of whole virus or live will also be found in these tests, but they won't distinguish between RNA only or RNA and intact virus. So it's important to determine that what we're seeing is just exposure to the virus. And many people, as we know, the large majority of people will either have very minor symptoms or maybe asymptomatic. So that's why testing is so important, not just for that individual, but also to look at the potential for widespread dissemination of the virus. As I said, they may be helpful with determining the extent of exposition if you do a quantitative test, but they don't determine if a person will develop serious life-threatening illness. We also don't really know if there is a certain number of virus particles that are required. What the limit of well, we do know what the limit of detection is, but we don't know how many virus particles it will take to develop a significant infection. And just a little note that I, along with a group of others through the International Standards Organization, ISO, are currently involved in the development of an international ISO standard trying to standardize the methods for collection of samples, because you can have a wonderful test, and if the sampling is poor, you might as well not bother. And standardizing the nucleic acid amplification testing procedures, because those have been all over the place, primarily because of the very short time span that has occurred between the beginning of this pandemic and even now. We've only been a year. Think about how long it might normally take both to develop vaccines as well as to develop tests that will withstand the significant quality processes to make them effective. There are also developing some laboratory antigen tests. So these look for protein fragments of the virus in respiratory samples. They tend to be what we call line immunoassays. So they develop an antibody to those protein fragments. They put them on a little strip, little like a pregnancy test, the antibodies, and then you spit or the material from the nasopharyngeal or the throat swab is put onto that strip and run. They're very rapid, so you can if they're positive, they can indicate the presence of active infectious virus, and they can be used for self-testing, point of care, and rapid test reporting, and they can occur in about 15 minutes. The problem is that there may be false negatives. They're less sensitive than PCR tests. So you're talking about micro or nanograms as opposed to picogram quantities. So they're much less sensitive by the order of maybe 10 to at least 100-fold, if not more. And false positives can also occur where there's disrupted non-viable virus. So those kinds of conditions have to be things, but they may be useful as we go along for looking for from a surveillance perspective. We can also do serology, the presence of specific antibodies to SARS-CoV-2 indicating infection that has occurred with an antibody response. And there's been at least 100 serology tests across the world that have been developed. The response has been quite variable. They're generally not as strong a response as was observed in the Mrna virus outbreak. And for example, in that outbreak, it was shown that if you had a virus antibody titer of about 1 in 640, that you could use that that serum from patients who had responded as a treatment for persons who'd acquired the MERS outbreak, and fairly successfully. So you were mopping up virus with plasma, sorry, with serum. It doesn't seem to have been that case. There are a number of studies going on in various parts of the world with this one looking for antibody responses for serum, which has high titers that can be used for these, and there are not many available. So again, the majority of people who are exposed to the virus have tended not, the active virus have tended not to make large amounts, significantly large amounts of antibody. And we also don't know how long lasting that antibody will be based on an active infection. And we still don't know because it's been too soon. We don't know if antibodies produced against the various vaccines will be long-lived. Again, there's also the innate immunity issue, so or the memory component of this, and so we don't know if the vaccines or if response to a virus will result in sufficient memory that if exposed again, we'll see lack of infection. So I want to turn in the last little bit to vaccines. So there are a number of different ways of doing this. An activated whole organism, live attenuated virus. And we see this both in viral and bacterial vaccines. Vector delivered, usually either by an adenovirus or another non-infectious vesicular stomatitis virus. And these have been shown to be good carriers, as it were. There are both mRNA or messenger RNA vaccines. There are small RNA vaccines. These are starting to be developed because they can be delivered into small areas for cancer immunization. And they're small, they consist of anywhere from 50 to 500 nucleotides, which is pretty small. There are protein subunits, and we've seen these most commonly in vaccines to Haemophilus influenzae and pertussis, and they're subunit vaccines, so they're not whole bacteria, there's the subunit components. Virus-like particles, which can be developed that look like the virus but are not infectious, and then DNA viruses. So the first ones off the block for this one have both been mRNA vaccines. So they're not infectious because they don't have infectious components of the virus. They don't integrate into the host. The mRNA is eventually degraded in the host, so they have pretty high safety. They're well tolerated. They've been shown to give a reliable immune response, and you can make them rapidly because they can be made cell-free. So you just take the nucleotide sequences and you just multiply them. So this is the genome of the virus. It's approximately 30 kilobytes. There's the spike protein. This is the E membrane protein. This is the nucleoprotein section. And this is the ORF, the open reading frame 1B protein. And the two ones that have been made to test both for PCR to test are the E membrane and the RDRP, which occurs in this region of the virus. And the genome is pretty small, actually. It's only 30 kilobytes. So what's happening with vaccines? So the first one off the block was the Pfizer that linked with BioNTech. This is an mRNA vaccine that targets the spike protein. The Mrna protein was the next one, a very similar one, also targeting mRNA, targeting the spike protein. One that's just been in the news lately comes out of Oxford, a couple Oxford University linked with AstraZeneca pharmaceutical company. This is a non-replicating DNA sequence for spike protein. So it's a DNA sequence virus or delivered via chimp non-infectious adenovirus vector. Sorry. Okay. So trying to create viruses that will have relatively, or vaccines that will have relatively be non-complicated for humans using the chimp as well, and a non-infectious vector to take the vaccine to the appropriate immune-producing cells in the host. The J&J one, which has just been licensed in the United States, is a genetically altered, non-infectious human adenovirus vector for the spike protein, and it's an mRNA vaccine as well. The Sputnik 5 in Russia, which we don't know a lot about, but appears to be fairly successful, is similar to the J&J and AstraZeneca ones. And they're all reporting relatively high success rates. There are a couple of others that are in various stages of development. Novavax and Sanofi GSK are using both the same procedure, which is a coronavirus protein generated in insect cells and then delivered with an adjuvant. And an adjuvant can be all sorts of things. For example, for Mycobacterium tuberculosis, we use adjuvants to deliver BCG to persons. There's a Medicago GSK, this is GlaxoSmithKline, which is a UK company. They're virus-like particles that are produced in plant bioreactors. So they actually look like the whole virus. If you see a photograph, I'm sorry, I don't have one on in here, but they're non-infectious. So they're basically there to compete with the virus for sites, but are non-infectious. They're all over the place. So the delivery depends on the supplier. So they can be either 1 or 2 doses. The timing has varied between 3 or 4 weeks for the 2-dose vaccines. And I'm getting my second dose about 5.5 weeks of the Pfizer vaccine, about 5.5 weeks after the first dose. Those are based on primary studies. And so I think we'll start to see differences as we move along. They're starting to get some evidence that that one dose with most of these may actually be sufficient to prevent serious infection and to prevent acquisition of the virus in somewhere between 80 and 90% of people. So they've actually done an incredible job in a very short space of time. The storage of the viruses has been controversial. It probably depends on initial determinations, making changes to the carriers, because all of these have to have some kind of carrier for them, or their vector requirements, which is actually another type of carrier. If you go back to pertussis vaccine, for example, the carrier and some of the other carriers, for example, are merthiolate and other components that may actually be important. And I think that they've tried to learn things from those so that not to create problems with adverse effects that are generally a result of the carriers, because the mRNA, for example, is a very small molecule. So the Pfizer is very cold, -70 to -80 degrees, but they're now looking at -20. And so it looks as though they won't have to have these ultra low conditions for storage. The Mrna is already a similar vaccine is being stored at -20, and the AstraZeneca and J&J ones, because they have these other vectors, can be stored in a refrigerator. And that makes a big difference in terms of your ability to do this. So how does antibody develop with these vaccines? mRNA is carried to the host cells in a lipoprotein, usually in a lipoprotein nanoparticle or vector. Sorry, that should say vector. With instructions to produce primarily spike protein. Okay. The host recognizes the spike protein as non-self, produces antibodies so that if there's only the spike protein there and nothing else, so antibodies will get produced so that when the virus appears, and we know that, as we've discussed, that the spike protein is the critical entry point for the cells, into the cells for the virus, antibodies are present to prevent entry and replication. And so the primary function of these vaccines is to prevent significant and serious disease. And the current vaccines, anywhere from 70 to 95% at preventing serious infection. They all appear to be very good in elderly populations. And basically, finally, the issue that's turned up, because this virus has spread so widely and in such concentration, undoubtedly other variants will occur. And we see this from influenza in our annual antibody drifts, where, so in influenza, as you may know, influenza occurs predominantly with what are known as drifts, and those are minor changes to either the neuraminidase or the hemagglutinin, and the vaccines are relatively able to mitigate against significant disease from those minor variants. When we see shifts in influenza, you get a major change, and that's resulted. So the swine influenza was a major shift in terms of those primary virulence proteins. Nobody had seen them before, and that's when you see this significant and widespread disease with them. So just to put all of that into context. So the coronaviruses constitute a large family of viruses that constantly mutate over time, like many other viruses. And the variants result from minor changes in the spike protein. And that's what we're seeing. And those current variants, this is just the virus trying to make its way in the world, like all pathogens. So those current variants appear to spread more widely, and we don't understand fully why. They appear to be more contagious in non-infected persons. The good news is that the current vaccines, at least in initial trials, appear to be relatively effective in preventing at least serious disease with these variants. There are three major ones now. This B117 that's been in the United Kingdom, and in Alberta, where we've seen a large uptick, in a fairly significant uptick in the numbers of cases of the variant here. And that's only because we're letting people back into the country. And then they spread it around because they don't follow appropriate precautions. It's not rocket science. There's the B1351 from South Africa that's caused quite significant disease there, but we've hardly seen any, probably because there's not much travel this way. And the P1 in Brazil, I don't know whether we've seen any specific cases here, but Brazil's in a mess. They basically did what Sweden did, didn't lock anything down. And so they have widespread disease in their own country. And that's when these variants are likely to appear. Because the virus is just, it just happens. There are mutation rates in the virus that will just occur. So there's some other factors which will mitigate against ending the pandemic soon. Vaccine hesitancy, social media, things like the blood clot issue that stopped the AstraZeneca one for the moment in all of Canada. There have been, as far as we know, only 1 or 2 deaths. Nobody's saying anything about the fact that influenza vaccine has problems with some individuals. One individual I know, for example, had significant issues with Guillain-Barré syndrome, which is known to occur with a very small percentage of patients who get influenza vaccine. Social media says they were approved too quickly, so they're not as effective, as efficacious as were indicated. There's no evidence to support that. Or that herd immunity is going to save us. So immunized persons don't continue to follow prevention methods. And right here we've got the several religious things, and I put that at the bottom too. So there's pressure to open up within countries. Travel, which is an economic issue. And as we know, these variants can move quickly. And the anti-maskers, they don't believe in simple measures that can prevent spread. It's to me, as a clinical microbiologist, it's absolutely mind-boggling, but it is what it is. So if you want a little note from me, it's do the right thing, prevent it from yourself and spread to your loved ones and your family, and you'll be okay. And then politics, big, big, big problem. And religion. So fighting among people and religion that presumably some God will save you. So we don't have to mask. We can put large numbers of people together just like a marketplace, and we will have cases. And then they will spread to others. And that's the biggest problem. So just to summarize, sorry I've gone on a little long, there will be other potential pandemics. The important things are early recognition and global transparency. So the virus spread is a result of levels of concentration in the population. And today, more than ever, rapid travel can result in that kind of spread. So we need to have accurate sampling of affected person, and we need good testing methodologies, tracking and quarantine. They're essential to reduce viral loads in our communities. So we also need have new technologies for rapidly developing and testing safe and effective vaccines as a result of these pandemics. The new technologies have represented a new paradigm for pandemic control, morbidity and mortality. And so this one we haven't had one since a real good one since if you want to put it that way, since 1918-19, when we didn't have access to those kinds of technologies. And so this has been a really good, very rapid response to trying to deal in a reasonable period of time with these issues. And then finally, continuing education and prevention, debunking the conspiracy theories, telling that individual who's not wearing his mask in the grocery store, hopefully he doesn't plow you in the face, that he's not helping reduce the environmental concentrations and supporting our actual long-term herd immunity. And that will stop the virus. I think that's it. So there were other questions. These are some of the things that I've seen, but I'll stop there and ask if there are questions from our group.
So any questions?
Yeah, I did have a couple questions to ask. So for the real-time PCR, is it always true that whenever you're doing the positive testing that the cycle threshold should always be less than 40 for a person to be tested positive for COVID?
Yeah, I think it depends on the test, but people are using a variety of things. So we're not suggesting that I think the limits of detection are more important, and it may depend on the type of thing, but certainly if we hear, we've used less than 35 to, so like other PCR tests, when the crossing points, the CTs get above, approach that 35 to 40, they will get retested. So it may be due to other factors that you have a high one. So there may be inhibitors involved. That's a reasonable, if it's less than 40, it's more likely to be positive than otherwise. But there's no real good specific number that every test can follow. So that's why the people running the tests look at every one of them. I mean, if they're down around 20 and you have good clinical information, it's positive. That's not a problem. It's when you get clinical information that tends to support and you have a higher crossing threshold that you need to be more careful. So I wouldn't exactly say that you can put a specific number on it, but that's reasonably close. If you say if it's more than 40, it's undoubtedly negative.
Other questions?
Again, sorry, just one more thing. Again, that's why a number of the PCRs are looking at more than one target as well, right? To make sure that they're looking at the correct ones. So it's just a further confirmation that you're dealing with this particular virus. Does that answer the question?
Yes, it did. Thanks. Thank you.
Yeah, other questions?
Yeah, I was wondering, has the expedited process of developing these COVID vaccines changed how other vaccines are being developed?
That's an excellent question. I know that there are other vaccines. There's a vaccine that's being developed. There's one vaccine being developed by our Nobel laureate, Dr. Houghton, over in Lai Kai-shing. And I don't know where that's at the moment, but that may well, he's been very good at developing hepatitis C vaccines and other things. It's quite possible that they will use that kind of expedited process to try and, depending on how things go. I know there's another one that's being investigated locally that is looking at an sRNA vaccine for target. That has some interesting things, most probably because of the small molecule size, the likely safety, pretty good efficacy. And I don't know, that one is at an earlier stage. the biggest thing that's happened in Canada, unfortunately, is up until now, the federal government didn't get on the ball to get production facilities going. So there's a production facility in Calgary now that can produce vaccines at large numbers of vaccines. And there were others. Canada had a very good process way back when through Connaught Laboratories for developing influenza vaccines. And as diseases changed, and I think people just didn't think, gee, we need to maintain so it's not that you can't develop them, it's that you can't make enough of them. And that's why we've run into some of these difficulties, because the vaccine producers just don't have the facilities to make the hundreds and hundreds of millions of vaccine doses that are necessary under the GMP conditions that are necessary.
Thanks for adding the clarification to those other issues too with production.
So speaking of the efficacy, I was just also wondering about how, like, for example, the mRNA-based vaccines, the Moderna and the Pfizer, they're all based in like the clinical trials and everything was done in the United States. But then there are several cities and their mayors, for example, in Detroit, that they banned the Johnson & Johnson. And they were basing that because of the efficacy. They were saying the J&J vaccines, they have lower efficacy compared to the mRNA vaccines. But then I was just wondering, and kind of that confused me, because I thought that the DNA is not as fragile as the RNA. And, since the J&J.
That's the problem, right? It's the conspiracy theories, I think. Yeah, so what's the difference between 80% and 90%? I mean, maybe you've got a different group of individuals in your trials. And it's the politicians who are making those kinds of statements without any science behind them. I hate to say it, but there is no pandemic. Just ask Donald Trump, right? Who didn't listen to the science. So that's the mayor of Detroit. What can I say?
So basically then, can the efficacies be compared to different vaccines? Because I thought different clinical trials based on the people, like there could be some other confounding variables.
Absolutely. I think that's right. But, let's think about influenza vaccine, that nobody asks that question. Influenza vaccine is probably 60%. All of these appear to be higher. Nobody says anything about influenza vaccine. We know it's not all that wonderful, but it helps to prevent the serious morbidity and mortality.
With aerosols,
sorry, I put that up there. That was
yes. What I'm thinking, Bob, is it depends upon the age of the building. Like probably in ECHA, air handling is better in the building than it is in the hospital, because the hospital was built a lot longer ago.
Well, the hospital, but the hospital has been changed. All of that interstitial space has undergone huge changes.
I see.
And so, yeah, the HVAC in the hospital is at high levels of code, right? Okay. Don't forget we have a major transplant unit in the hospital.
Yeah.
We have a public health laboratory in the hospital. And so all of those components are connected. I mean, I do accreditations for Food Inspection Agency laboratories and fisheries and oceans labs from the federal government. And I go into the, most of the accreditors don't, but I do, because that's important to the health of the workers in those places. No different than the hospitals. And so the hospitals our hospitals are accredited. They have to meet the standards for numbers of air changes, for filtration, for all of those other things that are code at this point in time.
Yeah.
So it's only places like, oh, I don't know, and I'm not. This is not saying maybe a big abattoir, for example, which is old, has been there forever. That's just an example. Please don't use that. Who knows whether they're required to do that or not? Yeah. They should be, but that's what's happened with some of these places, and so their air handling has been poor, and that was part of the reason for the lockdowns.
Yeah.
Even some gymnasiums, you know.
Yeah.
That nobody asks them about that. You got to put a lot of people in a gymnasium or a hot room in a yoga thing, and if the aerosols are, if the air handling is poor, the aerosol transmission or transmission, the likelihood is probably much greater.
Yeah.
So
okay.
It doesn't necessarily mean the age of the building. It means what's been done to deal with that. That shouldn't matter whether there's a pandemic or not.
Yeah. Okay.
The other thing that's, you know, there's been a lot of controversy, just quickly with the aerosol, is how does this virus transmit? Does it transmit by droplet, or does it transmit by, quote, aerosol? And there are other viruses and bacteria, for example, or anthrax. And we talked about that one before, right? Anthrax is a very small 2-micron thing. Your respiratory tract and my respiratory tract will kick out very small particles. This is a very small virus. So how big does the virus have to be contained in to be spread? Or do you have to have such huge numbers of virus particles in that transmission that are, quote, dry, as it were, that it doesn't matter? And so that's again why the ventilation is important. And all this modeling that's being done, I saw some photographs of huge things, and I mean, it's all some mathematician's thing. We need to have better use testing mechanisms for a lot of these things. I was thinking about that the other night. You know. Or people will run with these unsubstantiated issues. Right?
Yeah.
That next question that I had there, the long haulers, I don't know why. There's obviously issues in their immune functions, their own immune functions. Why do people get anosmia? Some people get anosmia. Both if that curler I was talking about and his wife, both of them developed anosmia. And anosmia in the curlers was about 20 to 30%. Hadn't been thought of before, earlier on in the pandemic. You know, and then the other issues are treatments. So there are immune modulators, like, as I told you, there are, what's the word I'm looking for? Oh, the spike protein seems to bind to the angiotensin converting enzyme. There are studies looking at modulators for angiotensins as an immune modulating mechanism, basically to bind or to modulate that receptor so that the virus can't impinge. Fascinating, cool stuff. Nobody really knows whether vitamin D is going to work. There are a number of studies going on. There are a number of antivirals, one that was used for Ebola, that are being tested in individuals with very severe disease. And those numbers will increase as people sort of search for possible ways to prevent death, serious illness and death in patients who are hospitalized and on ventilators. And then the third or fourth wave, it will depend on you and me, I think.
Yep.
And our ability to both get a vaccine that will have an effect, because we're not seeing any cases we're not seeing new outbreaks. The interesting thing, we're not seeing new outbreaks in our long-term care facilities because they've all been immunized now. So those kinds of results say that both immunization as well as, you know, the other things I talked about, masking, social distancing, all of those things make a difference and can prevent this.
Okay, well, that's a great teaching session, Bob. Thank you very much.
Okay, I found a number of extra typos that I didn't realize. I will send this back to you,
okay,
so that the students can have it if they want.
Yeah, great. All right, well, that's wonderful. So students, on Thursday, there is no class, but at the time of class, I'll send you the midterm. And then I need it back by Tuesday the 6th at noon. So you have a long time to work on it, as well as the feedback form, the Google form for your course evaluation. Yeah. All right. Very good then. Okay, thank you.
Was that helpful,
yes.
student?
Yes.
No, I'm not asking you. I want to know from them.
Yes, they're saying thank you. So
okay. Okay.
Yeah.
Well, if they have other questions, they can get hold of me through my email.
Right. Sounds good.
You can share that with them. Okay.
Okay, we'll do.
I'm sure it will change by tomorrow too. You know, never changing.
Yeah.